Yay! I think we might be in business here, folks. Anybody hear me? Hear me out there? Well, that was fun. Nothing like starting your webinar with a little bit of technical nightmares to get us going. So I hope everybody can hear me okay. We have a little bit different setup because we have trouble with the one we typically use. Whatever, roll with it. It's all good. So today, let's start again. My name is Eric Malice. I am the Chief Product Officer, and I'm also the co-founder here at National Test Systems. I have over 18 years experience, and I've always had a drive and a thirst for a better understanding, a better knowledge of all things clinical toxicology, especially as it relates to what we do here, to drug testing, and that's the important part. So what we do is we have a series of ongoing webinars. This one is probably 6 or so, if I'm counting correctly. And we bring these to you to bring you knowledge. We want to give you a better understanding, a better insight. We want to make more of me's so you can be out there and answer the questions. Really hoping to make your daily drug testing duties a little easier, or at least a little bit more insightful. I'm gonna lower my heart rate now. So what are we talking about today? Today we're going to discuss understanding the limitations of drug testing. We're going to discuss the good, we're going to discuss the bad, and we're going to discuss the ugly. Now, listen, I know not everybody wants to share the bad or the ugly with you. I mean, it's just the way it is. We want to all talk puppies and rainbows. But to me and to us as a company, it's much more important that you understand these limitations. It's just going to make your life so much easier if you have a better understanding of what a drug test can do and what a drug test cannot do. It's as simple as that. They're not perfect, but they're great. So this isn't going to be all doom and gloom. Don't get me wrong. There is the good in here. Don't miss that part. So what I did today to break this down for us, to make it a little easier to digest, I broke it down into 3 categories. We're going to talk about the qualitative, and the qualitative would be why something is positive when you expect it to be negative, why something is negative when you expect it to be positive, something from that range. We're also going to talk about the quantitative, and this is really mainly related to laboratory testing. And that's where we're going to talk about levels, about concentrations, about what they mean, about what they don't mean, and about what you can really hope to understand by having that information. And then lastly, we're going to talk about what I call the speculative, which I'm sorry to say, but a lot of that is going to be based on misinformation. Listen, there's a lot of information out there, and not a lot of it's good. I mean, you read on the news, on your social media. I read incorrect articles all the time from proper journalistic publications that just get some facts wrong. So you really, you have to be aware of where you're getting your information. And that's why I'm stepping. And I want to give you the best information possible. And I think I'm well equipped to do so. So we're going to clear up a lot of those speculative misunderstandings. So first, let's talk about the qualitative. Again, why something might be positive when you want it to be negative, or vice versa. So I will start off by saying this: the weakest part of an instant drug test is setting the cutoff level precisely. So I don't know how many of you looked at my blog post last month regarding poppy seeds. Some of you may have noticed that some of the positives that I got, that level was below the cutoff level. Now listen, our catalog is vetted better, and we have a higher standard than any other product out there. However, you're still going to see a positive where the cutoff is below, where the level is below cutoff, and you may occasionally see a negative where the level is above cutoff. It's typically very close to that cutoff, but it's important to note that sometimes there's just not enough of that substance in there to create a positive reaction. And that's on the primary drug. And when we talk about primary drug, we're talking about the calibrator. The calibrator is the substance, or the facet of the substance, whether it be a metabolite, a byproduct, whatever it is. The calibrator is what they set the cutoff level to on the test. So when we talk about the cutoff level not being great or not being perfect, we're talking mainly about the calibrator, or the primary substance that's targeted. Now, when we get into the secondary or tertiary substances, that goes down a little bit. The numbers that they list as reactive for those substances it's going to have even more of a variance. But let me get a little more specific for you. I'm going to use benzodiazepines as an example. So an instant drug test, I think most of them on the market these days, will detect 21 different benzodiazepines. Granted, there are 62 different benzodiazepines, so you're not going to get them all. So what the manufacturer does when they set the cutoff level for the calibrator on the benzodiazepine is they pick oxazepam. Now, oxazepam is also a benzo, but more than anything, it is the metabolite of many other benzos. So what we have is we have a test that's able to detect 21 different benzos, not just the target, but some of the ones further down the list, maybe the diazepam, maybe the alprazolam. Those cutoff levels are going to have a little bit more variance than that target drug or the oxazepam. So it's important to note that. Another good example on the same benzo panel is because we want to have the broadest test to detect as many benzos as possible, we can lose some specificity of some benzos. In this case, on benzodiazepine tests, you're never going to reliably get a positive benzo test from clonazepam use or Klonopin use. Now, we do have that, and that is definitely available as a test. But if they wanted to make the benzodiazepine test do that, it would make it no longer sensitive to all the other benzos that we really want to catch. So it's important to keep in mind what's being detected, at what level it's being detected, and how precise that level is going to be. Now, another thing is you can find all this information, by the way, on the product insert. The product insert is a multi-page fold-out that comes in every box of tests. There's a section toward the end called Analytical Specificity, and there it breaks down each substance, what will cross or what will react with that substance, and at what threshold. So all these numbers I'm talking about here, that's all in your box. Everybody's got one. Just take some time and read it. Some good information in there. The other reason why we might not get the result we're expecting is specimen concentration or specimen dilution. Now, I know I'm going to hit on this at least one more time before we're done here today, but this is huge. People have come to me and said, well, the guy admitted to using this, blah, blah, blah. And then if you look closely at the specimen, you'll notice it's a very diluted specimen. We check the creatinine level on the adulteration panel, and we see, well, yes, it's a very diluted specimen. So its chances of there being enough of that substance in the urine to cause a positive reaction, it shrinks with dilution. I mean, you can, if you went to the adulterant webinar, I spent a lot of time on it there as well. And then the final reason why things typically will or will not show up, in this case will show up on an instant test, is cross-reaction. So I mentioned the product insert and how it lists everything that can react. And we talked about the benzo, where it picks up 21 different substances. Now, technically, only the oxazepam, because that's the calibrator on the benzo, only that is a real honest reaction. All the other benzos that we're glad they react, but those are technically a cross reaction. However, when we really talk about cross reaction in the real world, what we mean is a substance that has zero relation to our target drug, and that is somehow causing a reaction that it shouldn't. So I'm going to give you the big three now. These are ones that are proven time and again. These are false positives, are more likely to happen than not happen. The first one would be Ritalin. Zantac or methylphenidate. And that is a medication for acid reflux disease. That's almost always going to cause a false positive on the methamphetamine panel. It just, listen, the tests have gotten a lot better and a lot of the false reactions have been worked out. This just seems to be one that over the past 30 some odd years, they have not been able to tweak that one quite out. So keep that in mind. That's going to happen. The other really common cross-reactant, proven cross-reactant, would be on the methadone panel, and that would be for doxylamine. Doxylamine is it's a sleepy part of Unisom and NyQuil, so it's a very common over-the-counter medication. Especially in cold and flu season, we often see a big spike in people calling about methadone false positives, and typically that's what happens there. And now the newest one to our list would be on the fentanyl panel, and that would be a drug called Buspirone, or brand name is Buspar or Buspar. Again, that's going to pretty consistently give a false positive on that fentanyl panel. And again, they always are working to make these things better. They've come so far in the quality and how quick they can get a new substance out. And we're always working hard on the R&D. So hopefully we'll see some progress on those three. But in the meantime, definitely a good 3 to know. And again, all that information is also in the product insert. Okay, next we're going to move on to the quantitative. There we go. On the quantitative, again, this is really geared toward laboratory testing. People who've talked to me on the phone or have dealt with me in other ways know I kind of have a little bug up my butt about people having the need to know level. And sometimes I overdo it. I get it. However, there's a lot of reason why I am like that. First of all, when we're talking about level, there's so many factors involved that a lot of times the level of the drug is not really giving you any more information than you had before you had that knowledge. A couple of different reasons. So the most common is about half the substances that we are testing for that are drugs of abuse, they don't come in a measurable dose. You know, they're not made in a pretty pharmaceutical laboratory. They're made in a bathtub. They're grown in a closet. They're grown in a warehouse. There's no, serving size, serving information on the size that tells you exactly the potency of what you're taking. You just don't know it. Also, a lot of drugs, depending on how they're ingested, any drug that's smoked, for example, it's going to be completely variable on how much of dosage you get with each inhalation, for example. How much smoke did you inhale into your lungs? How long did you hold that smoke in your lungs? The longer that happens, the more of the active ingredient gets into your bloodstream. So let's use an example. 2 guys share a joint on a Friday night, and you check their level Monday. The chances of those 2 levels being the same or even close is pretty slim to none. And if it were, it would be purely coincidental. And another factor that's huge in this is inconsistent metabolization. Not everybody metabolizes drugs the same way, via the same process, or at the same rate. So giving the same, 2 guys that smoked a joint together, we look at the levels and we see, hey, that's way different than one another. How did that happen? Well, we can point to the way the drug was used. We can point to the way they metabolize drugs as 2 big factors. Another factor is inconsistent metabolization, not just across people, but across time. If you look at the graphic that's up now, that's kind of how the drugs leave your system. It's not going to be a smooth slope from the top to the bottom. There's going to be peaks and valleys along the way. So one day you might have a spike in your concentration in your urine. It doesn't mean new use. It just simply means that happened to be one of the days where you metabolized more for whatever reason. Maybe you got a better night's sleep. Maybe you ate a healthier meal, whatever it might be. You metabolized differently that day, causing that level, again, to not really tell you what you need to do. And then the third thing, I told you I'd get to this at least one more time. If you do decide to look at a level, don't dare look at a level without also looking at the creatinine level. Creatinine is a naturally occurring byproduct in human urine, and it has a pretty normal range. If somebody has, again, somebody's level looks like it went down tremendously, but it turns out their specimen is 10 times as diluted, then no, the level didn't go down at all. So it's important, if you don't know how diluted or how concentrated that specimen is, by checking the adulterants for creatinine especially, then you won't know. And remember, while this is focusing on laboratory, concentration and dilution is huge on an instant test too. Everybody should consider testing adulterants, whether they keep it built into their cup or as a separate single panel of adulterants. It's not that expensive for the amount of information it gives. It's really something worth considering. All right, and then we're going to move to the last section here, which is called speculative. And again, this is more, misconceptions or just some vague things, why things don't always work out the way we hope they do. The first one on the list is the right drug or the right drug class. Now, this is huge. When we first started talking seriously about fentanyl, 2-3 years ago, we very, very frequently heard people say, well, no, I'm not worried about fentanyl. I'm testing opiates already, and it would be detected under the opiate panel. And it was a little worrisome that people were not aware, again, of the limitation of that opiate panel. It can't detect something that it's not made to detect. You don't know that you have a fentanyl problem if you're not testing fentanyl. You're going to use the right tool here. So it's important to know what each panel will detect so you know if you're getting a positive, what could have caused that positive. And if you're expecting a positive, why isn't it positive? A lot of that has to do with knowing what the test will detect, whether you have the right drug, or whether you have the right drug class. Another thing I like to call hard targets. Listen, if you see the list to the left there are 500 plus drugs of abuse that people just really don't test for. You have to find the balance between, testing everything that is happening around you and spending $10,000 ahead to get everything tested. But there's a lot of common drugs of abuse that people do not test for as a rule of thumb. Ritalin is still a very popular drug of abuse and probably going to be part of the topic of my next webinar. We see that spike a lot. People don't test for Demerol. They don't test for Special K, GHB. There's a lot of things that are available, but people, again, you got to find that happy medium. We want to test as much as we can. We don't want to spend $10,000 ahead to test everything. And then that kind of segues into my next point here is there's just a lot of substances out there. There are a lot of drugs of abuse. There's over 200 synthetic cathetone. There's over 100 synthetic cannabinoids. There's different families of speed and amphetamines that number in the hundreds. This is a lot of substances out there. So it's important, again, to know what's going on in your neighborhood. Follow the news, or more importantly, give me a call. I follow all of this stuff so I can bring it to you and hopefully slightly easier to digest packages so you can know what to be on the lookout for and know if it really is important. And then the final on this one is some misconceptions about specimen type. So I'll give you two examples of this. I've had a situation where a counselor tested one of their clients. Client was positive. Client denied it. Client said, I'm going to go to the hospital and get another test. So they go to the hospital and they get a blood test. So the gentleman calls me with this information. I said, I'm sure the blood test was negative. He's like, yeah, how did you know? Well, because the urine test is going to give you a picture of drugs you've done on the preceding day, 2 days, 3 days, further, depending on the drug. Whereas a blood test is only going to tell you what's in the bloodstream at that time, what they're under the influence of at that time. It's the same difference between a breathalyzer and an EPG test. It's just two different specimen types. It's not going to give you the same information. And I'll take that a step further. Hair follicles becoming very popular. Hair follicle testing is great. It goes back 90 days. It's impossible to adulterate. The problem is it doesn't detect drugs after use for at least 7 days. So we have people coming into programs, getting a hair test. Hey, your hair test is clean. Meanwhile, they've been smoking pot for the past 7 days. They're not going to detect it. It takes at least a week for that hair to grow out far enough. So again, it's important to look at the specimen type when trying to make the determination if you're going to get a positive or if you're going to get a negative. Unfortunately, that's just the way it is. Nature of human chemistry, as it were. So to summarize here, again, it's not all doom and gloom. I hope I shared some helpful insight. Drug testing is an invaluable tool. You guys couldn't get through the day without it, I would imagine. I wouldn't expect a construction guy to show up on site without a hammer. It's not the way you do your job. It's kind of impossible that way. The other good news is the technology is always moving forward. 20 years ago, we had problems where there was a cross-reactant on a test. It took them 10 years to sort it out. Or a new drug came on the market or came on the scene, and it took 5 to 7 years to get a test for it. We have a kratom test now, and it's only really been in the news for about 3 years. That's incredible. We have a working kratom test after 3 years. It's great. So yes, technology is way more advanced than it was in the past, and take advantage of it. This is a great time to be alive as far as technology, especially as this technology. And then lastly, let us help. Call me. Call your consultants. We want you to have as much information as you have. That is probably the biggest goal of this company, is to make sure you guys understand everything, know everything. It's just going to make your daily drug testing duties that much easier, and it's certainly that much more insightful. All right, so I see we've got some questions here, and if I can figure out how to work the computer I'm using right now, I will get to them. All right, I got a first question from Joshua Curtis. In my opinion, what does the future of drug testing look like? In my opinion, urine is almost always going to be the best substance to do drug testing. The best specimen type to do drug testing, just because of the nature of human chemistry. You get a picture of what somebody's done in the preceding days. when we come out with newfangled stuff like sweat patches, and the newest thing I heard is this new ocular scan that's essentially a lie detector. It might be helpful in some cases, but I think really the future is going to stick with urine as a specimen type, probably still going to stick with what's called immunoassay, which is the way the test works, chemical reaction. I do see improvements in the way we are able to report or record our results. Just today, or just last week, I'm sorry, we've looked at a scanner that you put the test cup in after you've run it, and it'll scan the cup, read the results, file the results, and put it right to your patient file. So in the immediate future, I still think that the first things we're really going to see that are going to be useful are going to be ways to better record our results. Next, we've got one from Ron Williams. Since busbarone is known to cross-react on the fentanyl, if we are sure someone is taking busbarone, can we be sure that it is the positive? That's a good question. Unfortunately, no. They don't compete in that way. So in other words, somebody could be on busbarone and have taken fentanyl, and the result would look the same on the instant test. So especially when we're dealing with these known cross-reactants, it is very important to still utilize a laboratory confirmation to really get to the bottom of what's there. That's a good question, Ron. Thank you. Next, we come to Amy DeHeld. Can you help me understand dilute UAs? I have criminal justice clients that often have dilute due to drinking much water, and probation classifies it as a positive drug screen. Creates a lot of frustration for the clients. Yes, I imagine it would. There's sometimes, as with everything, somebody's a little too quick to say that's bad, and somebody on the other side is a little too quick to say that can't be bad. The answer, unfortunately, is probably somewhere in between, maybe. However, I will say this is probably the most important part. Most of the legal entities we deal with, drug courts, probation and parole, that sort of thing, they consider that 20 milligram per deciliter creatinine, for example, as a hard cutoff. So they say, okay, if there's 20.1 there, that's fine. That's a good specimen. If it's 19.9, no, that specimen is diluted. Well, the real question is, sorry, not the real question, but the real answer is, if you've got a specimen that's 20 milligrams per deciliter on the creatinine level, it's pretty diluted already. Taking that a step further, if this particular individual is more regularly in the 100 and the normal 100, 120 range, then it's even more, it stands out even more as something that you should keep an eye on, maybe even doubt the validity of the specimen, even if it may not meet the true definition by being under that, what seems pretty arbitrary, that 20 milligram per deciliter amount. Thanks, Amy, that was a good one. Next, we have Ron Williams. Another question is gabapentin. We use Gab dips, and the dip's sometimes positive. Sometimes we send to the lab, and it says negative. Why is this the case? Is it cross-reacting, and if so, with what? So gabapentin test is very new. So yes, we might run into things from time to time that will surprise us. However, it's starting to look like NSAIDs, non-steroidal anti-inflammatory drugs. And that's just your good old Aleve and Motrin and acetaminophen. Typically, only in higher doses, those can cause a false positive on the gabapentin. However, since it has not been able to be reproduced with any reliability in a laboratory, it's not been listed on the documentation or the product insert yet, but it is something that they are working very hard on. Again, it is still a very new substance, so so sometimes we want to get it out there, but we also want to make sure it works, so we try and find that happy medium. As far as the gabapentin, probably what we're going to do, if anyone's experiencing this, is our next step is going to be to raise the cutoff level for the gabapentin. Right now it's at 1,000. Most of the gabapentin positives that I see when they get confirmed from the laboratory are in the tens of thousands range. So I think it's pretty safe that if we go to 2,000 on the cutoff level for the gabapentin, we're still going to catch all the gabapentin. Let's see. Can dilutes be caused by caffeine, or is it the amount of liquid ingested? For the most part, it is the amount of liquid ingested. Caffeine, for the most part, a diuretic. So if anything, it's going to make you, urinate more frequently. So the caffeine by itself does not have a direct result on the creatinine level. Something that would be a heavy workout, but that usually increases creatinine level. And so if somebody wants to really try and drink a lot of water and get away with it, they could go do heavy lifting for an hour before they do it, because that muscle waste is what the creatinine really is. And all right, it looks like we were able to get to all the questions today. If anybody thinks of something 2 seconds from now, because that's what always happens to me, give me a call, email me, reach out to your consultant, reach out to anybody here. If you want to schedule a call for individual training on something like this or another subject. Yep, I just got notified there are a couple more questions. This is why I don't use this computer usually. Do you have any idea on the medications that will cause a false positive for oxy? Also, is there any test we can use to test someone who's abusing Imodium? I'm going to do this one backwards. The Imodium, the opiate that's in Imodium is a fairly unique one. And it's a very, very low level. So I don't know of anybody who has or is working on an Imodium test. However, if somebody is abusing Imodium, by the way, if anyone doesn't know, Imodium is to slow down your bowels, basically. It's an anti-diarrhea medication, basically. So anyway, and what opiates do more than anything do really well is they cause constipation. So that's why there's an opiate in an over-the-counter diarrhea medication, if anyone's wondering. So anyway, because it's a very mild opioid and it's very small dosage, it shouldn't. However, if somebody is abusing it to get high, there could be enough, depending on the opiate. You know what? Off the top of my head, I don't know which opiate is in Imodium. I'll have this question logged, Missy, and I'll make sure someone gets back to you as far as which opiate it is, so I know if it will react at all on the opioid panel. That's an excellent question. The first half of your question, is there any medications that will cause false positive for oxy? Naloxone and naltrexone can, or I should say may. If the threshold for them to create a positive on the oxy panel is very high. And those are typically not drugs of abuse per se. People typically are taking known and prescribed doses of those medications. So while it can happen, it should happen pretty infrequently. If you start to see it on any sort of regular basis, let us know, and we can work with the manufacturer to see if that can be tweaked out a bit further. Next question from Selena. If a person drinks around 5 to 6 energy drinks a day, would that ever show up as positive for methamphetamine? Absolutely should not. The only thing I could think of is if somehow they got an energy drink that contained some sort of ephedrine or pseudoephedrine, that might turn positive on the amphetamine panel. But we've not seen any of those, guarana or ginseng or any of those other energy supplements cause any false positives on any of the panels up to date. A lot of those things are kind of black market and shady. So, if it's one of the real brands, I especially would be very suspect if it turned positive. Next, Charlie, we sometimes see pink sediment in urine specimens that are collected in the field. These specimens are rarely positive for adulterants. Any suggestions as to what we were seeing? Now, on that one, I would have to do some research. My gut feeling tells me it is possibly a medical condition, but if you're seeing it frequently, then I'd want to do some research, and that's an excellent. You've given me a puzzle, and nothing I like more. So, Charlie, we have your email will be on your question, and I make sure I get to it this week and have someone get back to you on that, because now I'm curious. I've not heard specifically of pink sediment. I've seen some strange colored urines, but there's usually something will show up as an adulterant when we measure it. So, Chris Montgomery, what about the cross reactivity of NSAIDs on the gabapentin panel? Somebody asked that question a little bit ago. It's still not been proven. It's still not able to be reliably reproduced in a laboratory setting, Chris. So unfortunately, it's